I'm speaking with Karla Kirkegaard, a professor of microbiology and immunology at Stanford University Medical Center, soon to be at Scripps, Florida. Thanks for joining me today, Karla.
Thanks for having me, Vince.
This interview is part of a series we're doing for the new edition of our textbook, and this one with you has to do with RNA synthesis. So I'd like to talk a little bit about your work in that area, but first I'd like to know about you a little bit, where you were born and educated.
I grew up in Fresno, California, and started college at UC Irvine. After a little bit, I transferred to UC Berkeley, where I changed my major a few times, as maybe some of our listeners have as well. I started grad school at Berkeley, actually, in the lab of Jim Wong, the discoverer of DNA topoisomerases. We'll probably get back to that later when we talk about mentorship. But anyway, I was in the lab for a year, and then he said, "We're moving the lab." I said, "Where?" He said, I don't know yet." So about a month later, he said, "You're going to have to transfer to Harvard," which was both exciting and terrifying for somebody who'd grown up in Fresno. But it ended up being fun, moving the lab across the country. Mm-hmm. Anyway, I graduated from Harvard, and I worked on the mechanism of gyrase and type 1 topoisomerases too, and then did a postdoc with David Baltimore. That's when I first met you, was when I interviewed for that. And I remember you were working, it's off topic, but I remember you were working very hard to very loud music. Anyway, I got my postdoc, I finished my postdoc there and went on to start my own lab.
I'd like to know how you got interested in a career in science to begin with.
Well, that's interesting because a couple of my majors in college had to do with literature and writing. But I always liked science, just very basic science, just trying to figure out how things worked and the clean logic of it. So I thought, well, I'll become a science writer. So that was my ambition, was to become a science writer. That's why I decided to get a PhD. But then I just found out that I really liked the practice of it. I've always been a person who worked with my hands. I did a lot of sewing and, crafts and bicycle riding. And I just really liked the process of working with my hands and my head at the same time and making experiments go together with ideas. So I just found out that I liked it and was reasonably good at it. And that's a wonderful thing to discover when you're young, that you're actually good at something you like.
Were either of your parents scientists?
My father's a physician, and my mother's an artist. So I guess probably the curiosity about science came from my dad. But what I didn't like about medicine, although it's getting less so, is there's a certain recipe to the way you treat things and the way, and then there's a certain amount of grandiosity you have to affect in order for the patient to believe you're going to help them. And I really liked, really knowing what you were doing in a very humble sort of way.
A lot of work in your lab has been on studying viral RNA synthesis. I'm interested to know how you got interested in that particular topic.
Well, when I was in graduate school, working on DNA enzyme mechanisms, these incredible experiments came out of Tom Cech's lab, and in which we found out that RNA could have catalytic activity. So that at once solved the chicken and egg problem of evolution that some of us worried about, which is which came first, the information or the product. So DNA, is what's thought of as the repository of information, and proteins do a lot of the work, but that RNA, which could both do work, do catalysis, and have information, created the speculation that RNA was like the mother molecule, perhaps the origin of life. So my idea was, well, maybe the closest you can get to that now is an RNA virus. And a simple one, like a positive-strand virus, in which the RNA encodes the proteins, which then work on the RNA to make the negative strand, et cetera. It's the simplest possible RNAviral lifestyle.
So tell us a key experiment in your view that came from your lab having to do with RNA synthesis.
Well, as you probably remember, it was actually because of your work that we could define mutations in RNA genomes, right? So it was like a party. We got to invent RNA genetics, right? You make a lot of mutations in the DNA copy, and then you have this faithful representation in the RNA. So everybody made a whole lot of mutations, and they would give rise to defective viruses. And then you'd try to rescue those phenotypes by co-infecting with a functional virus, and a lot of times that didn't work. So there was a lot of discussion in the field that maybe all the viral proteins acted in cis. That is, they acted only on the genome that made them, and they didn't diffuse very far away. That would have a lot of implications for evolution, because if you're a lonely genome and you're going to start making mutations and making different proteins, maybe you want to keep them to yourself. So it's an attractive idea, but not rigorously tested, because if you have genetic information, an RNA genome, it's very difficult to make a mutation in a protein that encodes it without making a mutation in the RNA. So obviously a change in RNA structure or function, if indeed it has function other than encoding, could make a cis-acting defect. So we reasoned that the only way we could deal with that is to recode the RNA and then show that it was okay. So we put a bunch of stop codons throughout poliovirus RNA, showed that when you suppress those stop codons with the suppressor tRNA and put in tyrosines instead, the RNA was perfectly functional, the virus was fine. Then we could say, okay, what happens to the virus if you stop protein synthesis here? What about if you stop protein synthesis here? Can you rescue with a co-infecting virus? And indeed, 3 or 4 of the proteins acted in cis and could not be rescued. And the other ones, could be rescued by a co-infecting virus. And those were consistent. Those were the polymerase, those were the capsids. So that means that some products are shared in the cell by viruses and some are kept private. And that has a lot of influence in the way we think about now how RNA replication occurs. That is, you have an RNA genome, makes a few proteins that probably drag it to the membrane on which it's going to replicate, and those are private. And then there's solution biochemistry that goes on with some of the other viral proteins. So the evolutionary pressures, the selection pressures on different proteins from the virus are different. And genetic transmission is different, and that's informed a lot of my work since.
I remember those experiments very well. Tell us how they influenced the field.
Well, you'd probably be in a better position to comment on that than I. I think a lot of people still make assertions about proteins acting in cis without doing a test of whether the RNA structure is affected. And there continue to be some. But I think it's pretty accepted that the proteins that interact with membranes and thus tether the RNA genome to where it's going to replicate have a different sort of function from the transacting proteins, the ones that assemble in an architectural way from solution.
I have to say that before your experiment, I had that view that these proteins must only act in cis. And I think we were just ignoring the RNA structure. So this was a welcome addition to our understanding. I think it's very important. And since then, I have viewed genetic results very differently.
Oh, that's great. That's great. Well, me too, because I thought I thought the other way. I thought it was all RNA structure, because coming from DNA, in entomology lab, in which we thought a lot about DNA structure, I just thought, it's all RNA structure, and this whole bizarre phenomenon is going to go away. So I needed to do that experiment to really appreciate how primitive it is to be an RNA genome, make some proteins, and then keep them to yourself. That's actually a very rapid way to evolve.
Okay. Tell us about some other work from your lab on viral RNA synthesis.
One of the reasons I got interested in RNA synthesis started when I was a postdoc, which is, again, as a graduate student, I was very interested in biochemical mechanism. And I remember going to talk to David Baltimore, who's a very, very smart person. I told him about my interests. And he said, I've always wondered, there's this phenomenon of recombination in RNA genomes. And, you end up doing a co-infection and then recombinants come out. At that time, there was not really a way to nobody had physically characterized the RNA that came out. They just knew that there were genetic markers going in and separately going into the co-infection and coming out linked. And he said, I've never been really sure whether it's true or not, but it would be an interesting mechanism should it be true. So that just caught my attention immediately. I became really obsessed by this idea. And I had this idea that there would be recombination because I read the literature, I found it convincing. And a really nice paper came out from another lab describing it. So from Andy King's lab, and it was in England at the time. And showing that there was some physical basis for this recombination event. So then it became an attempt to set up genetic markers. And that's where your infectious cDNA came in, because we started making mutants and looking for phenotypes. So I thought once we started studying the mechanism that there would be, RNA breaking, ligating, topoisomerizing activities in the cytoplasm, and that was my goal, to find all that. So we did show that there was genetic recombination. We showed where the sites of crossover were. But then it turned out to be replicative. It was that old idea, Messelson-Stahl, back in the day, when they figured out the mechanism of DNA recombination, it was either going to be breaking, rejoining, or what they called copy choice, which is when you're copying one strand, during replication, you switch over to the other one. And darn if it didn't turn out to be the case, that was what happened in RNA viruses. That is, we found positive evidence for its dependence on RNA replication of the first marker, but not the second. So as though you had to replicate through the first one, and then you were free to cross over. So that was great. And then I realized that instead of spending the rest of my near future characterizing, breaking, rejoining, and topoisomerizing activities, I was going to have to learn about RNA replication. So the field of RNA recombination has become a bit of a cottage industry. And it seems to be found in all, if not most, if not all, positive-strand RNA viruses. Curiously, not in negative-strand viruses. And I think we have a lot to learn about that and how shielded the genomes are from each other.
It's hugely important for plus-strand viruses. As you know, all the vaccine-associated polioviruses that we excrete after being immunized are recombinants.
That's right.
And rhinoviruses now are recombining. It's really amazing. So I remember that paper very well. It was really beautiful. And our listeners should definitely read it. There is already a reference in the chapter, as it is.
Thank you. Thank you.
So what are the big unanswered questions pertaining to viral RNA synthesis?
Well, the architecture of the RNA replication complex is a very curious thing. All positive-strand RNA viruses replicate on membranes, which makes it very difficult for biochemists, but very interesting from a cell biology point of view. Most biochemists were trained in solution mechanisms. You make a bunch of assumptions to talk about a Michaelis constant, et cetera. That all goes out the window when you have everything immobilized on a flat surface. So this sort of surface biochemistry makes the rules different and very interesting. And since that's a lot of what goes on in the cell for cellular activities as well, I think that the rules of how much affinity do you need if you're in a planar surface to form a complex? If you have an iterative complex like that, how do you recruit other viral and host factors from the cytoplasm? What affinities are required? And what they call in immunology avidity when you have an oligomeric surface. I think, again, these are very primitive mechanisms that if you want high affinity, you just make a lot of the same protein and then you slather it over the surface. We found that the RNA-dependent RNA polymerase of poliovirus, for example, if left to its own devices in solution, will form large flat lattices. And that's such a nice topology to then put on a membrane that I guess once we found that, I became convinced that this kind of array is actually a surface on which RNA replicates, rather than how people want to say it, a lot of like balls coming together. And whether or not the whole array uses those kinds of polymerase oligomerization surfaces, clearly there are a lot of other proteins involved. I think the idea that it's more of a catalytic surface than a traditional sort of solution biochemistry thing brings up a lot of interesting ideas that are kind of new biochemistry. I like that.
Is the replication on a vesicle or membrane structure, is this unique to plus-strand RNA viruses?
Well, I think so. I mean, although surface replication is certainly going on inside a double-stranded RNA virus. So if you think about a double-stranded RNA virus that keeps its genome inside a capsid. I mean, that's definitely a surface. And we know that sometimes the polymerase is embedded. So that means we need to think about, I mean, like we do in genome synthesis, more like the nucleic acid threading through an immobilized polymerase than how you like to think about it, which is, oh, the polymerase is going to be like a little train, going along the track. Yeah. That's how you teach, children about polymerases. But in fact, they're usually immobilized in the cell, and then the nucleic acid threads through. So that's double-stranded RNA viruses that replicate inside a closed core. Negative-strand viruses, they're these large ribonucleoprotein complexes, and their topology and organization and association with subcellular structures is, a hotbed of research right now, and I'm very excited about how it's turning out.
So tell us how technology has changed over the years from the early days in DNA enzymology to the present. How has it changed, and how has that affected what you've done?
I met somebody when I first started grad school who got his PhD thesis for determining that the last nucleotide in tobacco mosaic virus was an A. So that's really sad when you think about the millions of sequences we have, and which becomes a bioinformatic problem.
I like to tell classes that it took me 1 year to sequence polio genome, which today would take an hour.
Yeah. Well, at least you got 7,440 nucleotides.
Not one, right?
Not one. Exactly. I mean, that's just crazy, right? But how, I think, of course, deep sequencing and sequencing has changed everything. But right now, I would say that the ability to do bioinformatic analysis of these things is actually the key to understanding the wealth of data that's coming out. So, to tell you the truth, when people ask me, when high school students or even college students ask me if they can work in my lab, which is, full of pathogens, and sometimes, of course, I love to have undergraduates in the lab, but if they're, mature enough to deal with a lot of pathogens. But if for younger students, I say, look, take computer science, learn bioinformatics. That's the first training you should get in modern biology.
Wow, that's good advice.
Yeah, well, and you can do it in the context of your high school, college program.
Yeah.
Then you really go right to the top of biological analysis.
Right.
I'm actually learning bioinformatics in not in secret, but in my lab. We all have sort of rudimentary. We have a group meeting in which we teach each other bioinformatics and follow an online course given by one of my colleagues. So.
Great. So the next question may sound as if I've already asked it, but you could view it slightly differently. Of everything you've done in science, what single thing are you most proud of? Well,
pride, as it doesn't really come into science in that way, because everything you learn is just more humbling. I can tell you what I'm most excited about right now.
Sure.
We've been trying to explore these interactions between viral proteins in the cell, and we just kept getting surprises. For example, so RNA viruses mutate at a rate of, 3 times 10 to the minus 4 mutations per round of replication. That's really high. So we're talking about polio here, so in a 7,400 nucleotide genome, that means that all the progeny on average have 2 mutations. And of course it's the Poisson around that. So how do these mutant genomes interact in the cell? And how do you work with mutant viruses? I had a rule in my lab for a while that no point mutations, we weren't ever going to work with point mutations because they reverted so fast. And I had a student actually who's from New Jersey, and Deborah Hope, and she came and she had a mutation in the polymerase that didn't revert. It lost its temperature-sensitive phenotype at a frequency of 1 in 10 to the minus 6. I said, that's because there's another mutation. Sequence it again. Poor thing. So she had to do that a couple times. And finally, in sort of New Jersey slang, she let me know that she wasn't going to do it anymore, and there was only one damn mutation in there. And she was right. So that was when we discovered that mutant proteins could dominantly inhibit the outgrowth of wild-type variant viruses. So there were the mutant polymerases, because polymerase is an oligomer, would assemble with other functional subunits, and too bad that there was a good genome in the cell. It couldn't grow out because it was dominantly inhibited by its parents, cousins, brothers, sisters. So then we got the idea, wait a minute, what about drug resistance? If you had a drug-resistant virus that was born in a cell, if the drug targeted an oligomeric structure to begin with, the products of that drug-resistant genome would assemble with other more functional subunits, and the assemblage would then be drug-susceptible. So it was an idea for how to target drugs so that the drug-susceptible genomes would be dominant and the drug-resistant ones would never grow out. That paper was published in 2005. Scott Crowder, a postdoc in the lab, was the first author. And since then, I'm kind of on a quest to change drug targeting in RNA viruses and in other mutable genomes. That is, I think instead of targeting the active sites of enzymes, of monomeric enzymes, which is of course what's the model for how you make an antiviral, that we should target oligomeric surfaces, not to disrupt them, but to hyperstabilize them. Because then that's how you make, so then imagine, you have a wild type genome in a cell that's drug susceptible. You have a drug that binds and it hyperstabilizes a polymerase oligomer, a capsid oligomer, and then you have a variant genome because it grew before the drug came, but there's drug resistance that pre-exists. It's making capsid proteins or polymerase proteins that don't bind to the drug. But too bad, because they're going to oligomerize with the ones that do, and the assemblage is going to be drug resistant. So we've been employing this starting with capsids, with poliovirus, with dengue virus, et cetera. And the drug-resistant viruses can be dominantly inhibited by the drug-susceptible ones. When you infect a mouse with poliovirus and treat the mouse with a compound called VO73, which is what's in clinical trials for post-poliovirus eradication, drug resistance does not emerge, period. I mean, we were astonished. And if you look at a more conventional drug target like the viral protease, there's this compound called repintravir, which was the hope of a hope for a while that it'd be a good antiviral. You put that in, it inhibits the 3C protease, but variants grow out very quickly, presumably because if you have a good protease and a bad protease in the same cell, the bad protease doesn't bother the good one. So paradoxically, I think that the drug targets that we should be thinking about for highly mutable genomes are the more complex ones, the oligomeric assemblages. Some of them aren't even enzymatic. But now you can screen for hyperstabilization compounds, which is what VO73 does with the capsid.
So it sounds like you've gotten some companies interested in this approach, or have you done this entirely on your own so far?
We've done this entirely on our own so far because, and I mean, our listeners understand this because they're students and they're students of genetics, but it is a bit of an abstract concept. It's also not a paradigm. So I guess one of the reasons I'm moving to Scripps, Florida, is because I've decided to really take this concept that started as a genetic idea, and just come up with example after example of what kinds of viral proteins are dominant, what we call dominant drug targets, that is, will suppress drug resistance, and what kinds are not. So what there is in this new facility is a lot of high throughput screening facilities, and also I'll have the wherewithal to really pursue this idea, because that's my goal, is to really make this abstract concept more intelligible and even intuitive for not only for positive-strand viruses, but other mutable viruses like negative-strand viruses, and even other polyploid situations. What about cancer, when you get amplification of part of a chromosome, right? And you get multiple copies of an oncogene, one of which might become drug resistant. It's a big problem in cancer. But it's still in the cell with, 50 copies of drug-susceptible oncogene encoding genes. So, maybe if you knew how to target it so that they were dominant inhibitors of that oncogene. I think any mutable polyploid situation is amenable to this kind of thought.
So speaking of our listeners, many are students, and many of them would love to hear your advice, especially those who want to go into a career in virology. Can you give them some words? In particular, you've had great mentors, Jim Wong and David Baltimore.
Yeah.
Can't get much better than that. Is that really important?
It's crucial. It's what makes it what makes you become yourself. It's very funny because students always ask, "What should I take?" And I usually start laughing. I said, "You're a grad student now. You take what you want." Because that's your only hope to contribute in any kind of original way, right? Is to take your own ideas and the ways in which you're unusual and be that way as much as you can. I mean, in a scholarly way as well as otherwise. So I guess I chose both my mentors by I just met, when I got to know them, intellectually, I just wanted to think like that. It was not more complicated. So I took physical biochemistry from Jim Wong as a graduate, as an undergraduate, actually. And the clarity of his thought, and he has a terrible memory, which is one of the reasons, mine isn't the best. So, so he'd always start every conversation with first principles and build up from there. And I just loved that. Always start with, what is the question? In the simplest possible formulation. What do we know? And then we'd build up from there. And I just thought that way of thinking was what I wanted to learn.
This is why it's so important to have great researchers teaching undergraduates. It doesn't happen all the time.
Right.
But they're the ones with the passion and the real understanding. It's coming from working with the material and knowing it inside out.
Sometimes when you know material inside out, you can actually make it simpler and boil it down to its concept as opposed to, a whole bunch of acronyms and pathways.
Yep. Karla, I want to thank you for spending time talking with me today. I've always viewed you as the apple of science. Think different.
Well, sometimes you don't have a choice, huh?
Thanks a lot.
Thank you.